Hello, everyone. My name is Nita Chosia, and my mentor is Dr. Fatima Raymond. And so my topic was about potential biomarkers that played in the process of nodule metastasis and early tongue cancer. So I wanted to give out like a little intro, so the epidemiology of oral cancer. So oral cancer is the 11th most common cancer worldwide, and in most ethnic groups, it's actually found more in men and women. Now, I don't know why there wasn't really any details said about that, but I just thought it was an interesting factor. However, I did find some correlation why that correlates with the risk factors I'll explain next in the slide. The annual mortality is about 145,000 deaths, and areas that have the highest cases of oral cancer is usually in South and Southeast Asia. So particularly India has one of the highest, and then other areas that are less developed. And 20 to 30% of early oral tongue carcinoma will have occult neck nodule metastasis. So that means your lymph nodes are swollen, which is important because it deals with it, why could be an important prognostic factor for that. So risk factors. One of it is the excessive use of smoking. Non-smokers are 3 times less likely to get it. And the reason is because heavy smokers can have gingivitis. And gingivitis is the inflammation of gums, which can ultimately cause oral cancer. And then another one is alcohol. So alcohol increases objects to pass through the oral mucosa. That's bad. We don't need that to happen because oral mucosa itself acts as a barrier to stop harmful toxins from coming in. So the fact that alcohol increases the permeability of it, it lets harmful toxins to pass through. So back to what I said about men, found more in men and women. In many different places worldwide, I noticed that usually men will have more effects for oral cancer because they'll smoke more and they'll drink more alcohol. So that could potentially be why in ethnic groups it's found more in men and than in women. So I wanted to talk about the intracellular signaling pathways found in oral cancer. So this figure shows different types of pathways affecting gene transcription. So this includes regulatory pathways like MAPK, inactivation of p53, growth factor signaling, angiogenesis, and carcinogenesis. One of the most important genes in p53 is responsible for suppressing tumors. So once p53 is inactivated, tumors are able to metastasize and to continue to grow since there's no apoptosis occurring. Now I'm going to talk about the preliminary results. So the whole point of this project was to figure out which biomarkers had potential in being most differential expressed in the metastasis of oral cancer, so it can either be upregulated or downregulated. In order to obtain a goal, tissue samples were collected from 50 patients in which a tissue microarray was used to compile these results, all obtained from Dr. Raymond's lab. All of these results and data led to the 3 biomarkers that are obtained, the most important 3. So that would be protoplanin, ecodherin, and beta-2 microglobulin. So I'm first going to start out with protoplanin. So protoplanin, also known as PDPN, it's a transmembrane receptor protein, and so increases cell migration and invasion with the help of CAFs that stand for cancer-associated fibroblasts. So CAFs increase the migration of neighborhooding cells, and this brings me to the next bullet point on how protoplanin is associated in the early development of the tumor because it can identify lesions that will turn into the oral cancer cells after metastasis. This deals with contact normalization because it associates with normal cells that affect the growth of nearby tumor cells in the early stages of oral squamous cell carcinoma, or OSCC. Now, I wanted to talk a little bit about the pathway of it, some background knowledge on this biomarker. So it's an interesting protein because It doesn't have actual domains that are intrinsic, so it interacts with functional groups from other proteins to affect cell behavior. So pKa and CDK5 will phosphorylate serine residues, and then ERM, which is a family of the certain proteins, they'll associate with other proteins in the tail of PDPN, ultimately activating protein kinases and the pathways involved, such as ROCK and LMK, as shown in this figure. This can lead to cell invasion and connects how potoplanin targets lesions in the early stages of OSCC. Certain compounds, like antibodies, can negatively affect the motility of OSCC cells by targeting potoplanin. Now this brings rise to results I found in two different literature papers, both concluding how potoplanin is downregulated. So in this graph above, it showed that potoplanin is downregulated because as the oral cancer cells are metastasized, lower levels of potoplanin was expressed. There was a huge increase in potoplanin expressed in normal cells, in which in the first graph it's labeled as poorly differentiated OSCC. In the second graph below, it shows that potoplanin was expressed in weak/moderate stages of OSCC, correlating to how it was still both downregulated. So the next biomarker I want to talk about is ecadherin. So ecadherin is a transmembrane protein, and it is a cell adhesion molecule that is found on the surface of epithelial cells, and it is dependent on calcium ions. So cell adhesion, it's important because it's regulated by different signaling pathways, such as tumorigenesis and the WENT signaling pathway. You know, this leads me to the next pathway, but I wanted to state how I said cell adhesion is important, because basically cells can decide what they want to do in their microenvironment. This can either be changing the cell shape or stopping proliferation. So these signals happen through the adherens junctions, regulating different signaling pathways such as MAPK and WENT. And I want to talk about the last bullet point, in which ecadherin is a tumor suppressor gene, so it slows down cell division, and this correlates with the invasiveness and metastasis of tumors. This brings me to the results I found in my in one of the literature papers about ecadherin dealing with the mRNA, and this graph shows that ecadherin slows down the number of cells dividing, so it downregulates as OSCC cancer cells metastasize, leading to lower levels of ecadherin. And the last biomarker I want to talk about is beta-2 microglobulin. So it's found in almost all nucleated cells and biological fluids. It regulates the immune system. It is expressed at a constant rate. So it's interesting. Beta-2 microglobulin is known as a housekeeping protein, so it's a part of a complex class 1 molecules. These cell surface proteins will associate with T cells that have antigenic peptides. Now, beta-2 microglobulin can either be tumor suppressing or it can be tumor promoting. So, because of that, it is a stimulating factor for different pathways, such as angiogenesis and ENT, leading to the metastasis of cancer cells, as shown in this figure. So, with beta-2 microglobulin, there weren't too many details about how it constantly was affected or could play into oral cancer, so I wanted to look at results from different papers to compare different cancers tested with beta-2 microglobulin and see if it was upregulated or downregulated. And what I came up with was I found this first graph in one of the papers, and this talks about the renal cell carcinoma, and it shows how beta-2 microglobulin and renal cancer was upregulated because higher levels of the serum at 2.6 milligrams per liter caused a reduction in cancer-specific survivals, in which you can see through the red dotted line. So it decreased compared to the blue line, which was the serum levels that was less than 2.6 milligrams per liter. And then in the first figures, and I'm sorry, sorry about that. Just don't know what. There we go. And then in the second figure, it shows that the correlation of white blood cells with the serum levels of beta-2 microglobulin in Hodgkin's lymphoma. So these talked about different white blood cells that were initiated. So once the mean of the serum was calculated, higher levels of beta-2 microglobulin was found in the initial count of the white blood cells. That was more than 50,000 cells per cubic milliliter. So overall, with beta-2 microglobulin, this showed that it was upregulated compared to the other two for ecadherin and podaplanin, and they were both downregulated. And so this brings me to my conclusions. Amongst the 20 biomarkers that were looked at, these three were chosen as they were more differentially expressed. Like I said, podaplanin and ecadherin were downregulated, while beta-2 microglobulin is upregulated. So after doing all this, I was very interested in learning more about it. So I feel like it'd be insightful to have like another clinical study where it show these main biomarkers together as a panel to show how well it can predict the metastasis of cancer in people. Cancer is one of the most like important things in our world. Like it's found everywhere, and sadly, like many people have this disease. Like my own father died from it. So this made me interested in looking into more types of oral cancers and the types that could of the biomarkers that could affect it. So I just wanted to thank you so much, and I appreciate it.